Hello, we're glad you've joined us for this live webinar, Knock Out Biologic Quant and Quality on a Tiny Sample. I'm Xavier Gutierrez of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Unchained Labs. If you'd like to learn more about our sponsor, please visit www.unchainedlabs.com. Now let's get started. You can pose questions to the speaker during the presentation while they're still fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. I now present today's speaker, Lisa Adamiak, PhD. Lisa Adamiak is an application scientist at Unchained Labs. She is responsible for supporting new product development for protein analytical tools by carrying out proof of concept studies. Lisa received her PhD in chemistry from UC San Diego, where she synthesized and characterized biological nanomaterials for diagnostic applications. Lisa will now begin her presentation.
Thank you, Xavier, and thank you all for attending this webinar today. So as many of you may be familiar with, analyzing biologics can be tricky. Sometimes pieces of data can tell a story one way, but can actually reveal a completely different story when using accurate measurements or other complementary techniques. So really break down those illusions when it comes to your data analysis. Whether you need to measure protein stability, get some accurate quantification, or just get some information about the quality of your sample, Unchained Labs has the right tools for the job. Today, I'm excited to talk about two of our products, Lunatic and Stunner. For all of your UV-Vis quantification needs, big or little, Lunatic has you covered. Big Lunatic uses a plate-based format, measuring up to 96 samples at a time in just 5 minutes. Little Lunatic, on the right here in the screen, uses a chip-based format that is ideal for labs that want to quickly and routinely measure protein concentration. Each of these platforms uses just 2 microliters of volume per sample. So how does this work? The unique plate consumable or the chip consumable makes it all possible. So the consumable uses a dual microcuvette system that is connected to an input well via a microfluidic channel, which you can see here in the diagram. The full UV-Vis spectrum is measured with 2 path length sizes, both 0.1 and 0.7 millimeter path lengths. This enables the wide dynamic absorbance range from 0.03 all the way to 275 OD. So really you can measure wide concentrations of your protein from 0.02 to 200 mg per mil. After simply dropping the sample into the input well with a pipette, the capillary forces will guide the sample into the channel as shown in step 2. This is where the sample will reside until the plate is loaded into the Lunatic instrument. Then the pump in Lunatic will apply negative pressure to carry the sample the rest of the way into the cuvettes as shown in step 3. And then Lunatic simply reads the sample. Let's take an up-close and personal view of the plate. So notice that each sample is separated by its own microfluidic channel. This means that there's no risk of cross-contamination between samples, and each well is only used a single time. Also, the plate can be left up to 2 hours unattended before measuring because of the protection that the plate consumable offers from evaporation. Each cuvette has a fixed optical path length, so you know that your concentration is on the money every time. Lunatic can measure the concentration of all kinds of samples, whether it be a protein or nucleic acid, and whether it's pure or a mixture. Lunatic has lots of different applications that can quantify the concentration of your sample, and we'll get into some of these in a little bit. So here's a list of the protein-specific applications on Lunatic. There are different types categorized by conventional or classical quantification, along with unmixed and standard curve applications. Unmixed applications can quantify your protein in the presence of other impurities, such as nucleic acids, salts, or components like imidazole, often used during the purification process of his tag proteins. Or if you're inclined to run colorimetric assays like the BCA, Bradford, or Lowry assay, the standard curve applications are right for you. So there's lots of great options. Now, because of the two path-length cuvettes, Lunatic can measure a large dynamic range from 0.02 to 200 mg of mil of protein using just 2 microliters of sample. For really high concentration IgGs, you can actually go higher than this, up to 250 mg per mil, with high throughput and reproducibility. The algorithm for this IgG unmixed application actually extrapolates the shape of the absorbance curve to be able to measure these concentrations accurately. So you can use less sample, eliminate error with manual dilutions, and you don't need to worry about cross-contamination. And all of this can be done with protein solutions that are less than 40 centipoise. The fixed path lengths in the consumable enable both high precision and accuracy. So the same samples were measured on both a microvolume reader with a motor-driven path length and the Lunatic consumable measuring or containing the fixed path length. The other microvolume reader shows a lot of variation, especially at the higher protein concentrations, whereas Lunatic shows pretty accurate and precise concentrations. Unlike other UV-Vis readers, Lunatic gives the option to perform a wavelength-specific background correction with the classic A280 application. So this background or turbidity correction enables better accuracy because it uses an algorithm that takes into account the light scattered at each wavelength. Now, if you're using a standard protocol, the single wavelength background correction method may be required. So this application is also available on Lunatic if you need it. So why do this turbidity background correction? Well, this turbidity or background correction is wavelength specific, meaning it takes into account Rayleigh scattering. Said in another way, shorter blue wavelengths are scattered more strongly than longer red wavelengths, and because proteins absorb at the lower or blue wavelengths, we account for this scatter to yield more precise and accurate quantifications of your protein molecule. And so you could have actually see this in the previous slide if I take a look back here. With the same sample analyzed with the turbidity correction, we actually obtain more accurate quantification at 2.8 mg per mil instead of a little bit higher at 3.1 mg per mil. Now, this doesn't only apply to proteins either. With nucleic acid quantification, the A260 application also uses the turbidity or background correction. There's a lot of variety when it comes to genomics quantification, and similar to the protein applications, Lunatic offers the ability to do classic, unmixed, or other standard curve type applications. So the unmix is also available to deconvolute the impurities. In these unmixed applications, common detergents and salts are recognized as impurities and subtracted from the DNA or RNA absorbance spectrum. The big advantage of the unmixed A260 application is that it can save a lot of time over other quantification techniques such as fluorescent dyes. So you just need to measure directly, and Lunatic doesn't require any dilution steps. For UV-Vis quantification of proteins or nucleic acids, Lunatic is really the only system out there that can measure your biologics at high throughput and high concentrations. It uses just 2 microliters of sample, and it takes just 5 minutes to measure up to 96 samples. You can tack on the unmixed applications, and Lunatic makes batch concentration measurements really a no-brainer. So now if we take the power of Lunatic and combine it with a technique known as dynamic light scattering, or DLS, we get Stunner. Now, Stunner is the first and only platform that combines UV vis for concentration and dynamic light scattering for sizing. Stunner not only has all the quantification power of Lunatic, it can also assess the particle size of your sample too. It also uses a plate-based format, just like Big Lunatic, measuring up to 96 samples at a time with just 2 microliters. So often when working with biologics, the first question that's often asked is, how much protein do I have in this sample? Usually followed by, is this sample still good? With Stunner, these questions can be answered in a single experiment in one shot. So let's say I'm in the lab and I want to check my new batch of protein, my batch of protein from last week, and then a really old batch, all of which were stored in the fridge. As I double check the concentrations, I really want to see if there are aggregates in the sample. However, I don't want to spend a lot of time doing size exclusion chromatography to assess the quality because it's time consuming. Stunner gives me the ability to perform DLS to check particle sizing. And here's an example of that. So if we compare my good sample, it's showing a z-average diameter of about 11 nanometers, which is the expected size for a monoclonal antibody. The PDI, or polydispersity index, which I'll describe later, shows a very monodisperse value, less than 0.1. And the concentration is close to what I expect. Now, if I take a look at my sample from last week, the z-average diameter is a little bit larger, and the PDI, or polydispersity index, is also a little bit larger, indicating that there's something maybe a little heterogeneous in the sample. And you can see that in this intensity distribution curve, that there's 2 peaks instead of just 1 in the sample. Now, both of these samples are the same concentration, but DLS, or dynamic light scattering, is able to give me information about the size of the particles in solution. And in some cases, it can contain several aggregated populations. So in this case, the sample that's really old in the fridge is really quite messy. Even though it's the concentration that I expect, the dynamic light scattering is showing me that the z-average is now very large, along with the PDI. And so this is indicating that there's more than one type of particle in solution. The Stunner plate is actually very similar in design to the Lunatic plate, in that the microfluidic channels are also used to connect the 2 pathlink cuvettes to the sample input well. However, the Stunner plate enables simultaneous UV vis and DLS measurements. So when DLS is measured with concentration, a full plate can be measured in just 1 hour. Stunner can also be integrated into robotic platforms for even higher throughput. And the DLS specs meet industry standards, measuring particle sizes anywhere from 0.3 to 1,000 nanometers. Now, some of you may have not used DLS before, so I want to go over the technique quickly. So dynamic light scattering, or DLS, measures particle diffusion, also commonly called Brownian, or referred to as Brownian motion. The sample is essentially irradiated with a laser, and particles moving in the solution will scatter the light in all directions. The detector will collect this scattered light signal over time. Now, all of this looks like random noise, but the rate of change of the signal, or the diffusion coefficient, is going to give us the information that will relate to the size of the particles in solution. So, take a small protein, for instance. It will move fairly quickly, and so the fluctuation in scatter will change over time fairly rapidly. And you can see this as the intensity of scattered light is plotted over time. It's kind of moving up and down fairly quickly. A larger particle, on the other hand, will move pretty slowly, and the intensity fluctuation actually reflect these slower movements. So if we just transpose that smaller particle intensity over time with the larger particle intensity over time, you can actually see if you start to compare these small time intervals, it's easier to notice rapid changes for the smaller particle than it is for the larger particle. You will essentially have to wait a longer amount of time to see a change in the intensity of light for a larger particle. Now, what do you do with all this information? So the way that you get size from this is that each sample has a unique scatter profile that's measured over time, which will then be used to give you a correlation function, shown here. Each curve has an associated diffusion coefficient when fitted to a single exponential. So that will give you the important information that you use to assess the particle size in a sample. So notice here that there's two correlation functions, one for a larger particle and one for a smaller particle. The larger particle produces a correlation function that takes a longer amount of time to decay, whereas the smaller particle's intensity scatters light more quickly, so it decays more quickly. So once the correlation function and the measured diffusion coefficient is obtained from that correlation function, the coefficient, the diffusion coefficient, is plugged into the Stokes-Einstein equation, shown here. So the Stokes-Einstein equation uses the viscosity and the refractive index of the solvent at some temperature to determine the hydrodynamic diameter of the particle, so essentially giving you the particle size in the sample. So the 2017 ISO standard is applied in Center to analyze all of this math for you so you can focus on getting the data that you need. So dynamic light scattering is a super sensitive technique that is really good at picking up on small changes or on the very tiny presence of very, very large aggregates. For example, if you take a mixture of particle sizes, 5 and 50 nanometer particles in diameter, the 50 nanometer particle diameter will scatter light 1,000,000 times more than the 5 nanometer particle. This is because the intensity of scattered light is proportional to the diameter to the 6th power. This makes DLS really an ideal technique to detect tiny amounts of large things. Now let's take a look at what a polydispersed sample looks like. So this is a sample with multiple populations of sizes in solution. One smaller, around the 8 nanometers in diameter for a protein, and a larger population of aggregates, around 270 nanometers. And you can see this shown in the intensity distribution. So when the cumulus analysis is applied to the correlation function shown on the left, the z-average diameter gives a size somewhere around 38 nanometers. Now, if you take a look at the mass distribution shown below in blue, you notice that the smaller peak, the 8 nanometer one for the protein, is the main contributor to the light scattered in the sample. However, this all comes back to the intensity of scattered light relationship to the diameter. So because of that diameter to the 6th power, you can see that the larger thing scatters much, much more light. So you have two peaks in the intensity distribution, whereas you have a single peak shown in the mass distribution. And you can kind of see this reflected in the correlation function. So you can definitely see that there's two populations kind of coming from inside the correlation shown by these green lines. And that's kind of reflected in the intensity distribution. So in center analysis software, both intensity and mass distribution curves are reported. So let's take a look at this further. The intensity weighted distribution indicates if there are aggregates in the sample based on the scattering of the sample. The mass distribution describes the relative proportion of multiple populations in the sample. So this is quite useful since the relative proportion of monomer and aggregates are known for a particular sample. As DLS is very sensitive to trace amount of aggregates, you can actually see in this particular case that the DLS can detect less than 1% of larger particles in solution. Stenner software is very streamlined. So it reports the absorbance spectrum and the dynamic light scattering results like the correlation function, intensity, and mass distributions that we've discussed before. And you can see all of this directly on the screen. You don't need to be a DLS expert to analyze the data. The results table indicates the concentration measured at A280 for proteins and the average hydrodynamic diameter shown as the Z-average diameter, along with the parameter known as polydispersity index, or PDI, as we kind of discussed before. So getting more insight into that, the PDI sheds light on the heterogeneity of the sample. So if a sample contains more mixtures of particle sizes, the PDI can become quite large, larger than 0.1 in value. For this particular protein, although the Z-average diameter shows approximately what you'd expect for a monoclonal antibody, the PDI is approaching 0.12, which indicates that there may be a small portion of aggregates in solution. Now, for long-term stability experiments, size exclusion chromatography, or SEC, is the method of choice to evaluate percentages of monomer and protein oligomers like dimers, trimers, or higher order molecular weight aggregates. However, it takes a long time to run samples by SEC, with additional time to equilibrate the columns and run standards. So in this example, we ran a monoclonal antibody on SEC that was stored in a salt formulation at 25 degrees over time. And we evaluated the sample at different time points by SEC. Now, Stenner is a pretty complementary technique to evaluate sample quality over time, especially when it comes to pre-screening samples prior to running SEC. So here, Stenner will likely pick up the higher order molecular weight species as they grow in over time, as you can see. Now, keep in mind that DLS has a resolution limit, so species like dimers and trimers are not necessarily likely to be picked up or detected by DLS. Nonetheless, the Stenner UV-Vis can also pick up concentration of your protein and fragments so that you can measure both quantity and quality as a quick check prior to running your SEC. As expected, looking at these same samples day to day on Stenner, Stenner was able to pick up the high order molecular weight species at the as shown by SEC. On day 0, the monoclonal antibody has an expected diameter of about 11 nanometers and a small PDI of less than 0.1, so that this indicates a monodispersed, pretty clean sample. We observed the Z-average over time not only increasing, so by day 6, day 14, and day 21, we can see this slow increase. We can also see that the PDI increases over time, indicating that multiple particle distributions are in the sample. Now, Stenner is pretty powerful because it can pick up on these small changes so that you know which samples you need to run on SEC today, or maybe the ones you can wait on and run SEC tomorrow. Not only does Stenner tell you the quality of the protein, it can also measure the concentration, which can indicate whether you're losing protein over time. So the measured UV-Vis spectra of each of these samples indicate that we're actually losing a bit of our protein as aggregates are forming and perhaps precipitating out from solution. Now, being able to measure both concentration and DLS simultaneously makes running colloidal stability experiments very easy. On Stunner, you can actually measure both B22, which is the second virial coefficient, and KD, the diffusion interaction parameter, using both static and dynamic light scattering modes, respectively. Both B22 and KD are actually independently measured values that describe weak, nonspecific intermolecular interactions of a protein in solution. By measuring a dilution series of a protein in a formulation, the B22 and KD values are obtained. And because the protein concentrations are measured at the same time, you actually get better accuracy for these values. So these values indicate whether a protein has net repulsive intermolecular interactions or net attractive interactions with other protein molecules in solution. This information can help guide you to identify better formulations for your biologic. So for example, in this as shown here, if the B22 is greater than 0, or has a slope greater than 0, it indicates repulsive forces for the protein molecules in solution. But if the B22 slope is less than 0, it can indicate attractive forces for the molecules in the solution. Measuring accurate concentrations for B22 and KD experiments is really key. Now, typically, all the concentration of all the dilutions need to be measured prior to running a DLS experiment to obtain these parameters, which can take a lot of time. Stunner, however, measures the concentration and DLS in a single experiment, saving you both time and ensuring greater accuracy. So if you look here, the B22 plot for a formulation screen of a therapeutic antibody was done in about an hour. So the formulation that has no excipients, or the formulation with sucrose, proved to be the best formulations for this monoclonal antibody. The formulation with arginine or salt, because they're a little bit less positive in the slope, are less optimal because of this relative magnitude of the B22 value. After an experiment is finished in the client software on Stunner, the analysis software opens to reveal your results. So the results from the B22 and KD experiments are shown and can be easily toggled between sample groups of different conditions. In addition, you can highlight each sample to look at the results, whether this be the absorbance spectrum, peak diameter, or the B22 or KD values. Now, all of these parameters are provided for you so that you don't have to do the complicated math to calculate them. Stunner software is very intuitive to use, so it's accessible to both DLS experts and novices alike. Whether it be a B22 and KD experiment or sizing and polydispersity experiment that you want to measure, your concentration and DLS results immediately opens, and it's clearly visualized in the Stunner analysis software. You can easily flip through each replicate in the tree mode view on the left panel and toggle between the absorbance or the DLS screens shown on the right. You can also customize your reports in the Report Settings tab at the top there to export exactly what you want to see. It's also convenient to select multiple replicates on screen for comparison. So if you want to take a glance at the absorbance or the intensity distribution for the whole plate, you can easily do this in the Overview tab, or also compare your replicates for each of your experiments. There's many ways to visualize your data. Stunner also offers the ability to make your own application with Homebrew. This is an exciting feature on Stunner, which enables more flexibility in the experimental setup and its analysis. And we'll talk about this in a little bit. So in the Settings tab for the Homebrew application, you have the option to measure any wavelength of interest, include your DLS measurements, and select a background correction method that best applies to your samples. So So here you can simply select whether you want to measure protein, DNA, or RNA. You can also choose your peak filters if you want to. So this is really useful if you have excipients in your sample that show particle sizes around 0.5 nanometers. And if you want to filter these out, you can increase the minimum size of that DLS filter from the default, which is 0.3 nanometers, up past 0.5 nanometers. So there's many different ways that you can actually select your settings that you want to measure your data. Now, by clicking the Output tab, you can create an equation, really any formula that you want, that will apply to your absorbance spectra. This could be concentration, which is supplied as a default formula, or average concentration and standard deviation. In this case, we're also interested in calculating the aggregation index, so we're using this particular equation. Stunner analysis will then use these equations to calculate aggregation index for our samples by using these values measured at A280 and A350 and using this formula. So as shown here, you can supply the units for your particular formula, and you can also identify the number of digits you want to see, whether it's significant digits or digits of precision. And we really have this calculator platform here to make it easy for you to identify the formula that you want. You can also preview what your data will look like right before you run the application. So if you decide you want to make some changes, it's easy to click the back button and define your experiments and the results exactly the way that you want. And notice here that the formulas that we put in the output list are shown directly in the results table, including the concentration. So really, in this example, we stress tested a monoclonal antibody that is sensitive to vortex mixing. So we then formulated this protein with and without detergent, PS80 shown in the list on the left, and we subjected it to mixing at different time points. The absorbance spectrum and DLS were both measured on Homebrew, and the aggregation index of the samples was calculated using the Homebrew app and reported in the table, just as we previewed it. So you can actually see here the aggregation index for this highlighted sample is reported below concentration, standard deviation, and then aggregation index. Plotting all the data together, we find that the more turbid the samples are, the higher the aggregation index. You can see that the formulation with the detergent, the PS80, was successfully able to stop aggregation at the different mixing time points, while the formulation without any detergent was not able to stop aggregation. So the aggregation index essentially peaked around 2 hours, as you can see in this graph. And what's really neat is that this was confirmed by DLS, which was run on the same experiment with the same set of samples. So if we look at this a little bit more closely, you can see that the formulation without the detergent PS80 showed a very large hydrodynamic diameter along with a very high PDI value, indicating that the sample was very polydispersed with aggregates present in solution. Now compare this to the formula with PS80, you can see that the sample remained pretty closely aligned with time zero for that same formulation. Overall, there's so many great options for your UV-Vis and DLS measurements with Stunner. Stunner has all of the cool applications that Lunatic has, but with the ability to assess particle quality with DLS. Whether you want to perform a quick DLS measurement or use a mix to deconvolute impurities in your sample, maybe check that your purification protocol is good, or maybe you want to brew your own custom applications with Homebrew. I mean, you can do all this in addition to measuring colloidal stability for the sample. So Stunner can pick up on all the small changes like this, so you really know that what you're measuring in the sample is what you want to see. Stunner software does the heavy lifting, so you don't have to. Everything is streamlined from the application screen, the wizard guided setup, and the data visualization so that everyone can really use it. For higher throughput needs, both Stunner and Lunatic can be used, and they could be fully integrated with liquid dispensing robots with options with API. The plate format for both Lunatic and Stunner are also compatible with any pipetting head. And for easy sample tracking, each Stunner plate and Lunatic plate are equipped with a barcode. So this is important for integrated systems that ensure traceability and detection of the wrong plate or maybe random plate loading. You can also add a 21 CFR Part 11 package to your Lunatic or Stunner software if you need to meet compliance. This add-on includes everything you need from full sample tracking, data authenticity, and user accountability with electronic signatures. So it includes features such as system and experiment-specific audit trails, electronic signatures like I just mentioned, and data integrity checks. So get the quantity and quality of your sample in the same experiment with Stunner. Stunner allows you to learn more about your biologic by measuring both concentration and dynamic light scattering on the same set of samples using just 2 microliters. You have the flexibility to get the information you need with sizing and concentration, colloidal stability experiments, or you can even brew your own application. Now with the right tools, getting accurate concentration measurements can be a great thing, but measuring accurate concentration and knowing whether your sample is still good can be a beautiful thing. Thank you all for tuning in, and I'd be happy to answer any questions you may have.
Thank you, Lisa, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window, labeled Ask a Question, and click on the Send button. Lisa will answer as many questions as time permits. Now, the first question is, what needs to be done to prepare samples for a B22KD experiment?
Yeah, so preparation for a B22KD experiment is actually pretty straightforward. All you need is your protein stock solution, which is typically less than 20 mg per mil for a B22KD experiment. So once you have your protein stock solution in hand, you can simply dilute that to make at least 5 other concentrations of protein. And then you would also measure your blank. And so what happens is the scattering intensity of that blank is subtracted from the samples so that you can get the scattering intensity of the protein itself. And so the Stunner analysis software will calculate the B22 values based on that information along with the scattering intensity of a known standard. So typically a DLS calibration standard needs to be run every 6 months, and that value will be used in the equation for B22. So aside from just measuring the calibration every 6 months, you can simply load your sample solutions, your dilutions, with your blank, and then hit go, and then you'll have your B22KD results. Now what's nice is that concentration is measured at the same time, so you don't have to actually measure the concentration before the experiment because it's collected simultaneously.
Excellent. And are both of the cuvettes used for making the measurements?
Yeah, so both of the cuvettes are used in the measurement. So the 2 cuvettes are the 0.1 millimeter path length cuvette, and the second one is a 0.7 millimeter path length cuvette. So the 2 cuvettes are both used in the UV-Vis measurement, but the second one, the 0.7 millimeter path length, is used for the DLS measurement.
And if I've already measured the UV vis of my samples, can I reanalyze the data with the equations I want?
Yeah, absolutely. So the Homebrew application on Stunner makes this really easy. So the Homebrew application is where you can actually enter your formulas. So the equations that you want to analyze your UV vis data in a particular way. And simply all you have to do is if you've already measured the UV vis, you can actually recalculate that data set with your own custom-made Homebrew application. So it's really easy to toggle your data set between recalculation to get the information that you need.
And what is the minimum protein concentration you need for a DLS measurement?
So pretty much across the board for all DLS systems, the minimum concentration is 0.1 mg per mil of lysozyme. And so for Stunner, this is the same. So you want to make sure you're at least at 0.1 mg per mil of lysozyme for DLS.
And if I want to measure samples in different percentages of glycerol, do I need to take the viscosity into account for DLS?
Yeah, so both the viscosity and the refractive index of the solvent play a part in the calculation of the sizing, the particle sizing of your sample. And so if you have, special solvents or unique excipients in there that can cause changes to the refractive index or viscosity quite drastically from that of water, then those values you may want to enter to make sure that you have accurate particle sizing. But Stunner makes this actually really easy for you in that you can enter all these values into preset analytes and buffers. And so simply, let's say you didn't know the value for your viscosity for a 30% glycerol sample, what you can do is measure the samples, measure the samples anyway, and then once you figure out those values, either from published references or you actually measure the value, you can recalculate the data set with the new viscosity value to get more accurate sizing results.
Excellent. And is this instrument equally as useful for crude samples, comparison being to Fort Bio?
Yeah, so this is a little bit different because Stunner is a standard UV vis reader. So there's no binding that's occurring within the chip like there would be with a Forte Bio instrument. However, we do have the unmix applications. So for like crude lysate samples, for example, you can use the unmix application on Stunner. So it's called the total protein application. And what this does is it actually subtracts other types of impurities away from your sample. So things like salts, detergents, azi, things of that nature, and nucleic acids. However, it won't separate a mixture of proteins within your sample. So all proteins will essentially absorb at A280 and will be reported together because there's no binding that's occurring on the chip.
And looks like we're going to have one last question. What is the dynamic range in comparison to the slope spectroscopy?
Yeah, so the slope spectroscopy is equal to the solo VPE. So we have two path lengths that enable a pretty good range for that. Our high end, for example, is 250 mg per mil, so it's pretty good with respect to the IgG.
Excellent. Well, I'd like to once again thank you, Lisa, for your presentation. And I'd also like to thank the audience for joining us today and for their interesting questions. We'd also like to thank our sponsor, Unchained Labs, for underwriting today's educational webcast. This webcast can be viewed on demand through July 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.